Hi, everyone. Welcome to the, I'll be letting people in as we're speaking. Welcome to the first in this season's BEAD seminar series. BEAD stands for Biostatistics, Epidemiology, and Data Management Core. And we provide research support services to faculty throughout the institution. And currently we provide 20 sponsored hours per fiscal year for faculty in the departments of medicine, pediatrics, gynecology and obstetrics, and also ACCM. And we also have a service line providing patient-reported outcomes support services for faculty throughout the institution. And if you are interested in accessing our services, we also provide fee-for-services for all faculty as well. And we support everything from study design to complex biostatistical analysis, grant writing, and REDCap, EPIC extractions, PMAP support, everything in between. So if you're interested in accessing our services, please email beadcore@jhmi.edu, and we will get back to you to walk you through the process of setting up a consultation with one of our leads. On that note, I want to introduce our speaker today. Ethan Gough is an assistant scientist in the Bloomberg School of Public Health Department of Nutrition and is also a faculty lead with the BEAD Core. And he is able to provide support not only in the area of microbiome research for us, but in a lot of other statistical areas as well. So we've been grateful to have him as part of our team. And we're all looking forward to hearing what he has to say today. Just as a note, there will be an evaluation coming out at some point after this. We would really appreciate your feedback. Also, if you have questions, Ethan has said, feel free to just unmute and let him know what the question is or put it in the chat. So without further ado, take it away, Ethan. Thank you so much.
Thanks, Laura. So I'll share my slides and so are you seeing my slides?
Yes.
Okay, great. So yes, as Laura said, my name is Ethan Gough, and today I'll be talking about the human microbiome. I'll just give an overview of sort of how microbiome investigations are conducted, covering the basics and some of the analytic approaches and analytic considerations. And then I'll sort of walk through an example using a real data set. Before I begin, maybe how many people tuning in have some experience with microbiome studies, microbiome data analysis, maybe just by maybe putting yes or no in the chat, just so I can have a sense. Okay, I see lots of no's, but a few yeses. And none but very curious. Okay, all right, so that's great then. So I do have some sort of background slides to kind of give an overview of what the microbiome is before I get into the nitty-gritty of the analytic considerations. So you've probably heard of the microbiome. Definitely, if you're here tuning in, you've definitely heard of it because the title of the talk had the word microbiome in it. Estimates suggest that coverage of microbiome research has increased by almost 16% per year between 2007 and 2019 in the popular media, popular press, keeping pace with PubMed publications, which have increased approximately 19% per year in that time. The catchy titles of these media stories, such as Tending the Body's Microbial Garden, The Importance of Infants' Exposure to Microorganisms, and Unlocking the Secrets of the Microbiome, reflect the fascinating science that has emerged and grown in that time, and also reflect our growing understanding of the role of the microbiome in health. Two of the major early initiatives that were undertaken to investigate the human microbiome that have really significantly contributed to our understanding and sort of fueled scientific interest in microbiome research have been the Human Microbiome Project, or HMP, and the metagenomics of the human intestinal tract, or MetaHit projects. So HMP was launched in 2007 in the United States with funding from the National Institutes of Health, and was completed in 2 phases, with phase 2 results published in 2019. The broad goal was to characterize the enormous number of microbes that live in and on the human body using non-culture-based techniques, so DNA sequencing, and to elucidate the role of these microbes in human health. Also in 2008, MetaHit was launched in the European Union with funding from the European Commission and was completed in 2012. It also had the broad goal to understand the role of the human microbiota in health and disease, but with a focus on the intestinal tract. And sort of the major MetaHit publication came out in 2010, describing the major findings of MetaHit in Nature. And then in 2012 and 2019, the sort of major findings of the first phase HMP and the second phase HMP projects were published also in Nature. Now, these studies and the huge increase in microbiome publications, microbiome research outputs in that time were sort of largely facilitated by or fueled by the huge decrease in sequencing costs around the beginning of 2008. So these data come from the National Institutes of Health. On the y-axis is the cost for sequencing 1 million base pairs of, or 1 million bases of DNA. The curve is on the natural log scale. So even though the y-axis is in raw dollar amounts, the curve actually reflects the sort of multiplicative decrease in the cost of sequencing starting around 2008, when many sequencing facilities switched to sort of the next generation shotgun sequencing platforms that were quite a bit cheaper. And so this decrease, orders of magnitude decrease in the cost of sequencing starting around 2008 and continuing up until 2021 has made it a lot more affordable to do these sorts of sequencing-based, non-culture-based microbiome studies compared to what it cost 2 decades ago. They're still quite expensive to do, but compared to 20 years ago, there's an enormous difference in how much more affordable these studies have become. So what is the microbiome? So one definition that I've seen and that I use, I think it's a fitting definition. The microbiome is the combined genetic material of all microorganisms at a given site. And it consists of a wide variety of microorganisms, including bacteria, archaea, viruses, fungi, protists, but is often used to refer to the bacterial component. So sometimes you'll see terms like virome or microbiome, which have been coined to refer specifically to the viral component of the microbiome or the fungal component of the microbiome, for example. But the word microbiome technically refers to all the microorganisms as well as their the genetic material that they encode. The microbiota is another commonly used term, and that generally refers to the organisms themselves, and sort of conceptually refers to the microorganisms. Doesn't really account for the genetic material that they encode, all the genetic material that they encode. Now, the microbiome also covers our entire body. The composition varies between body sites and between sublocations within individual body sites. Even body sites that were once thought to be sterile are now known to harbor a low biomass biomass microbiome, for example, the lungs and the breast milk. But by far the largest biomass and diversity of microorganisms is found in the human gastrointestinal tract, particularly the large intestine. And most of my microbiome research has focused on the gut microbiome, although more recently I've been begun to be involved in projects that investigate other body habitats like the airway or lung microbiome, the vaginal or female reproductive tract microbiome, and also the breast milk microbiome. Now, it has been estimated that in the human colon, where the largest biomass and biodiversity of microorganisms exists, it's been estimated there are as many as 10 times the number of microbial cells as in the entire human body. More recent estimates place this more at a 1-to-1 ratio of human cells to microbial cells, but that's still a very impressive number of microbes that inhabit our bodies. We're almost, or we're practically just as much microbes as we are humans. And 99% of genes in the human body are in these microbial cells, which has led the sort of microbiome research community to think of the microbiome as sort of the second genome because of the enormous metabolic potential encoded in these microorganisms that far outnumbers the metabolic processes that are encoded by our own bodies. Also, the microbiome is highly variable between individuals and even within the same individual over time. It also can change in response to diet, other perturbations like antibiotics, infections, inflammation. So this fact is a large part of the reason researchers are interested in understanding how these microbes impact our health, because the microbiome is quite a bit more pliable than the human genome and outnumbers the human genome. But how do we measure the microbiome? So this is a busy slide, but I'll sort of just walk through it. Here I've summarized like the major steps in measuring or quantifying a microbiome. So both of these, there are two sort of main approaches. Both of them start off with extracting DNA from a biospecimen. Are there questions, Laura? I feel like I see some movement on my screen, but nope, nothing. Okay. So yeah, there are two main approaches. There's a whole metagenome shotgun sequencing approach going down the sort of left side of the slide, and the amplicon or marker gene sequencing approach, which sort of goes across and down the right side of the slide. Both start off, like I said, with extracting DNA from a biospecimen. The DNA represented by these colored bars here are then sequenced, and the sequences are used to identify which microbes the DNA come from, and thus to get a count or a measure of how many of each different type of microbe is present in a biospecimen. Now, the difference between these two approaches is the amplicon or marker gene sequencing approach focuses on trying to sequence specific genes within these microbial genomes that can be used to identify the microbes that the genes come from. For microbiome, bacterial microbiome investigations, the gene that's usually targeted for sequencing is the 16S gene because it's present in all bacteria. And it has a highly conserved region, sort of represented by these red and yellow sort of circles that facilitate PCR amplification. but they also have highly variable regions that facilitate differentiating one microbe from the other, one bacteria from the other. And so the 16S approach then sort of ignores the rest of the genome and just tries to amplify these genes that are easy to amplify and that can be used to differentiate one microbe from the other. Those genes are sequenced, and then the sequenced genes, which are termed reads, are then they then go through a sort of processing or filtering phase where reads that are low quality, meaning the sequencing didn't work properly or well enough on those reads, those are removed. Reads that are too short are removed, and also reads that are sort of artificially produced by the recombination of unique 16S genes from two different bacteria to produce like a new sequence that appears to be a new organism, but isn't. It's actually an artifact of the PCR amplification process. Those are termed chimeras. Those are also removed. And then the reads that remain are binned based on similarity. So reads that are similar to each other are assumed to come from the same organism. So those are grouped together. The two major approaches to do that involve either binning reads that are 97% similar to each other in terms of the DNA sequence, or more. At least 97% is sort of the typical cutoff that's used. And those are assumed to come from the same organisms. Or another more recent approach is bins these reads into ASVs, or amplicon sequence variants, which rather than using a sort of arbitrarily selected level of similarity between reads to determine which reads come from or are likely to come from the same organism, this instead bins reads based on whether they observe similarity between them is more similar to what you would expect once you account for the sequencing errors that occur when the reads are sequenced on the sequencing machine. The number of reads in each bin are used to give you a count of or a measure of the abundance of each OTU or ASV that are present in a biospecimen. And once the bins are formed, you can classify the organism or microbe or bacteria represented by each bin by comparing a representative sequence to a reference database. Two commonly used reference databases are Green Genes and Silva. Now, some limitations of the 16S amplicon sequencing approach is that 16S gene is only found in bacteria. So other microbes, for example, this hypothetical green microbe here, or the DNA of this green microbe here, doesn't have the 16S gene, possibly because it's not a bacteria. At least this is attempting to be a visual representation of that. So this would be missed by 16S sequencing. Only bacteria would be picked up. Some microbes have a different number of copies of the 16S gene, like this one has 3, for example, which can result in this bacteria being identified a bit more. There are also primer biases where the amplification of 16S genes from certain species are not as efficient, or the amplification is not as efficient as for others. And that's sort of represented by this one here, where one of the sort of primer binding sites is a black circle rather than yellow, suggesting that it's different. And so that's not picked up in the sequencing either. But some also, there are other reasons why PCR can amplify some microbes better than others, leading to sort of skewed representations of which microbes are present in a specimen. But the amplicon sequencing is more affordable, it's cheaper because less sequencing is needed because you amplify the 16S gene prior to sequencing. Now, the other approach, whole metagenome or shotgun sequencing, also starts off by, again, extracting all the DNA from a specimen, but sort of random bits of the entire genomes of all the microorganisms are sequenced to try to identify which microbes are these genes come from, which microbes the DNA comes from. And so regardless of whether that random portion of the DNA of a microbe contains the marker gene or not, is still sequenced. This is more expensive because more sequencing is needed to sort of cover the full genomes, to get sort of representative coverage of genomes to identify these microorganisms. But once the sequencing is done, the reads are taken through a processing or filtering step where you remove DNA sequence from the host. So in the case of the human gut, you'll remove DNA that comes from the gut itself. You remove reads that are of low quality, too short reads as well. And then these genes can be classified to determine which microbes the genes come from by comparing them to a reference database. And two commonly used ones are the NCBI and the Integrated Microbial Genomes Database. Another advantage of whole genome sequencing is that because you're sequencing all the DNA, not just a marker gene, the genes can also be annotated against reference databases like KEGG or Metapsych that give you some idea of the sort of metabolic functions that the microbes are capable of. And so those then can be sort of grouped into metabolic pathways, metabolic enzymes, or other categories of gene groups or gene families. And that can be carried forward into your analyses, as well as your analyses of the compositions or the taxa. Going back to the 16S side, although the 16S gene approach, amplicon sequencing approach, doesn't directly allow you to measure functions because you're only sequencing the marker gene, there are tools available that can sort of help you sort of predict what the functional profiles or predict an approximate functional profile based on information about which microbes are present and using available reference genomes from those microbes to guess at what the metabolic functions or potential might be. So that was, I think this is my busiest slide, and it's a slide that I'll probably spend this is the slide that I'll spend the most time on. I think the other slides are going to be less of a mouthful. But just to summarize, whole metagenome sequencing, amplicon sequencing, those are the sort of two approaches. Whole metagenome sequencing is more expensive. You need to sequence millions of reads or hundreds of millions of megabase pairs of DNA to cover sufficient amounts of full microbial genomes. Whereas amplicon sequencing, you can get away with tens of thousands of reads. So it's a lot cheaper. However, whole genome sequencing is more comprehensive, covering not only bacteria, but archaea, fungi, viruses, and functions. Amplicon sequencing is less comprehensive. It covers only bacteria if your marker gene is 16S, or if you're interested in fungi or other eukaryotes, there are marker genes available. But there are marker genes that can be targeted for those. But again, those are only going to identify the eukaryotes. They're going to skip the bacteria. Approximate functional profiles can be predicted. Whole genome sequencing, I see there's a question. Just let me wrap up this slide. Whole metagenome sequencing can also allow you to classify microbes down to the level of species or even strains, whereas with the amplicon sequencing approach, oftentimes you're not able to get better than genus level resolution. Some species level resolution is possible, but in my experience, you still mostly end up with predominantly genus level resolution. And there are some sorts of bias with the amplicon sequencing approach that are not present with whole genome. So one question from Neil, with whole genome sequencing, how do we know the DNA is that of bacteria versus archaea versus fungus? So that just depends on the reference database you use to annotate the whole genome reads. So I've used Metaflan, for example, which is a pipeline, a widely used publicly available pipeline, bioinformatics pipeline for annotating and classifying whole genome sequence reads. And Metaflan comes with a reference database that was curated by the creators of that pipeline, and it includes, excuse me, reference genes from bacteria, fungi, and viruses, as well as protists, I believe. So yeah, it comes down to the reference database that you use.
Thanks, Ethan. So what about some of the new strains where we don't have a reference database? How do we know the DNA belongs to what category of organism?
Yeah, that's a good question. And so I should say that with these approaches, there's definitely going to be a lot of noise in the data that you end up with. For that rare reason, I mean, there's going to be species that are identified or sequenced that are not well represented in a reference database, and those reads could potentially be misclassified, or they could just be unclassified, and you oftentimes end up with a portion of your reads that are unclassifiable, and they're just kind of lumped together in an unclassified group. Another question from Jay here is, how common is horizontal gene transfer? So that does happen. I don't have a good sense or a good number for how often that happens in nature. But it is a source of sort of noise or bias in these sorts of studies. I know with Metaflan, which is the pipeline that I've used most often, they sort of try to map the distribution of reads that they think come from the distribution of reads that seem to come from the same organism, and any sort of and they expect that the distribution of reads is going to be uniform. So if all the reads come from the same organism, they should all be uniformly, or at least close to uniformly, present in a specimen, because they all come from that one organism. Any genes that map to that organism that sort of are outside of that sort of uniform distribution, they sort of flag as potential horizontal gene transfer events, and they sort of discard those. Several species classified as having multiple affiliations. I'm not sure what you mean by affiliations. Neil, could you clarify?
When we get the results.
Yeah, sorry, go ahead.
When we get the results, some of the species are not given specific names, and then they're also represented as having multiple affiliations. So as if there is like one species with multiple genus. So I don't know if this is like a horizontal gene transfer going on.
Yeah, I don't know what the specifically if horizontal gene transfer would be the explanation for that. But typically reads are annotated based on some sort of score that tells you how closely they match genes from a reference database, and anything that sort of doesn't meet that that's sort of minimum score to say, okay, we're confident that this matches the genes from a particular microorganism closely enough to say that it came from that microorganism, then it's usually not mapped to that microorganism. and any sort of ambiguous reads or end up being lumped into the unclassified group. Does that answer your question? Also, Jay, does that answer your question?
Still
Thanks. It
something.
does.
Okay, I think you said yes. I'm not sure.
Jay said yes. Neil was sorry. I may have cut Neil off.
Okay.
No, it's still something which is unclear because there tends to be adaptation of the microbiome according to the body site. So one of the questions I had is the possible change of microbes from like a epigenetic kind of change undergoing to adapt to that body site. Could this be referring as the horizontal gene transfer happening?
Yeah, maybe we could talk a bit more about that offline, Neil. Just so I can maybe get through the rest of the slides, but I'm happy to talk more about that with you offline, if that's okay.
Go ahead. Go ahead.
Okay, so now I'm gonna just kind of walk you through some analyses of a real data set. So I'll be using data, publicly available data set from this paper that was published in Nature a few years ago. The relative OTU abundances derived from 16S sequencing from this study, from these children, classified to the genus level, are available on the principal investigator's website. And it's a data set I've worked with before, so I just kind of pulled it for the purpose of this seminar. So on the left here, I have a heat map of taxon abundances, just to kind of give you a sense of, if you've never seen or worked with microbiome data before, just give you a sense of kind of what the distributions look like. So the darker colors represent more abundant taxa or genuses or genera, where each sort of column represents a unique taxon at the genus level. And the rows going across are represent individual specimens or infants or infant samples from the Bangladesh study. There were 448 specimens in that study. Most studies have much fewer specimens than this. And there were 152 unique genera identified. If we had data from whole genome sequencing annotated to the species level, for example, there would probably be quite a lot more than 152 unique taxa at the species level, which would probably put the number of species on par with the number of specimens, which is usually the case with microbiome studies. Usually the number of variables is equal to or more than the number of specimens. At high dimensional setting can create or does create challenges with data analysis. The data tend to be sparse in places. So in these areas where these sort of white columns here, those are taxa that are completely absent from some of these specimens. That creates analytic challenges as well. The distributions of these abundances tend to be skewed. And there's also something called compositional bias that we have to contend with when we're dealing with relative abundance data. So here are 10 randomly chosen taxon distributions. These are natural log transformed to try to make them a bit more normally distributed, but they're still quite skewed. So the Streptococcus and Escherichia shigella are sort of the most closely to normal of all the distributions here, but they're all quite skewed. And some of them are just, because there's so few of this particular genus present in these kids, we end up with these really sort of sparse distributions like this one and this one and this one, for example. Others are just sort of heavily skewed towards 0, and sort of have these sort of long tails representing the abundance in the few infants where they were detected. Now, what I mentioned, compositional bias is one other challenge with analyzing microbiome data. So what exactly do I mean by compositional bias? So what we get once we annotate these reads and sort of count the reads that are classified as coming from specific microbes, we get read counts. And typically what we do is that we'll sort of express these read counts as a fraction or proportion of the total number of reads that came from a particular person or specimen. So in this hypothetical example, I have hypothetical species A, B, C, D, E, F, G, with read counts for patient 1 of 50, 25, 10, 6, 0, meaning species E was undetected in patient 1. 8 and 12, the total number of reads is 111. And so each species is expressed, the relative abundance of each species is calculated to represent the proportion of all the things detected in this patient that's represented by each species. So species A represents 0.45 or 45% of the total things detected in patient 1, for example. This creates an analytic challenge because if we look, for example, at just the raw read count for Bifidobacterium and Lactobacillus, the distribution of these, there's a lot more Bifidobacterium in these infants than Lactobacillus. And what happens when we sort of normalize these counts to represent the proportion of the total that's represented by Bifidobacterium or Lactobacillus is that Bifidobacterium across the board makes up a big proportion of the whole, and Lactobacillus makes up a small proportion of the whole. And so when we scale these as proportions, we end up with, in most cases, high Bifidobacterium, much lower Lactobacillus. And it sort of induces these negative correlations that don't actually exist in the count data themselves. And so here is a correlation plot of the, or a plot of the correlation between Bifidobacteria and Lactobacillus on the sort of count scale. So I just draw your attention to this square. As you can see, the magnitude of correlation is super slow. You can barely even tell that there's a little circle here. But over here, if we look at the correlation between the relative abundance of Bifidobacterium and the relative abundance of Lactobacillus, there is actually quite a large negative correlation in the relative abundance scale. So some of the approaches that we use to deal with these analytic challenges, to deal with issues like the high dimensional nature of the data, the skewed data, many zeros, compositional bias, etc., is that we come up with these composite measures of the makeup of each microbiome that we borrow from ecology. We apply dimension reduction or ordination techniques, multiple hypothesis testing procedures. We often filter out rare taxa. So those that are present in less than 5–10% of specimens are oftentimes excluded from analyses. We apply data transformations to commonly advocated for transformations are something called the center log ratio transformation, and the other is the arcsine square root transformation. Or we can try non-parametric methods or more flexible models that don't require or assume that your data are going to be normally distributed, and that allow sort of mixtures of distributions where a portion of your data is continuous and some other portion of your data is represented by zeros, so to account for the sparsity. And then there are more advanced machine learning and feature selection techniques that I'm not going to talk about. I'm really just going to address the ones that are highlighted in bold. The first important ecological concept is the idea of alpha diversity. This is really a measure of the number of different types of things that are present in a sample, in this case, bacteria. There are several measures of alpha diversity, and here are 4 of the most common ones. Richness is really just the number of different species types that are present in a sample. It ranges from 0 to infinity. So here I've calculated the richness in my hypothetical species data set here. There are 1, 2, 3, 4, 5, 6 species detected in patient 1, so the richness is 6. Likewise, 4 that are detected in patient 2, so the richness is 4. Evenness is another important concept. It's a measure of the uniformity of abundance distribution. So it gives you a sense of whether a microbiome is dominated by just one or a few taxa, or whether the abundance of several taxa is fairly even in a specimen. The Shannon diversity is also commonly used. This measures the uncertainty in species abundances in a specimen, and is a measure of diversity in that it measures sort of the variability in species abundances. It ranges from 0 to infinity, but it places more weight on rare species. The Simpson index measures the probability that 2 randomly selected species are going to be different. It ranges from 0 to 1. It places more weight on dominant species. So it's generally a good idea. It's generally recommended to use more than one measure of alpha diversity because they sort of reflect different things and sort of capture different aspects of the makeup of the microbiome. Another important concept is beta diversity. So beta diversity measures how many different types of things, in this case, bacteria, 2 microbiomes have in common. And beta diversity is meant to measure the turnover of members of a microbiome, meaning the loss or gain of species or taxa across groups or across environmental gradients. So, in the human microbiome case, we would be looking, beta diversity would represent the loss or gain of microbiome members in exposed versus unexposed patients or cases versus controls. 2 common measures of beta diversity are the Jacquard and the Bray-Curtis. The Jacquard is sort of visual displayed down here. It really just measures of all the things identified in each patient. This hypothetical, in my hypothetical table here, 3 are only present in patient 1, and 1 is only present in patient 2, and 3 are present in both. So the amount of non-overlap between patient 1 and patient 2 represented by these 3 and this 1 is what the Jacquard index quantifies. And if we think of, say, hypothetically, patient 1 had some exposure and patient 2 did not, then analyzing this similarity between patient 1 and patient 2 gives us some idea of the species loss or gain, species turnover over the gradient of exposure. The Bray-Curtis is also commonly used. It takes into account the proportion or abundances of species, whereas the Jacquard index only looks at presence or absence. These indices are generally analyzed either visualized or analyzed using ordination methods. So those sort of range anywhere from principal components analysis to non-metric multidimensional scaling, which essentially all try to reduce the abundances of all the taxa in your data set to fewer variables that retain as much of the abundance information as possible, but that can be easily visualized and analyzed using multivariate analysis techniques. Or at the very least, they try to reduce the taxon abundances to fewer variables that at least preserve the differences between the microbiomes, even if they don't actually fully aim to retain the large portions of the variance in your data set. Then finally, we can just look at differential abundances, going sort of 1 taxon at a time. We can just compare the abundance of 1 taxon to the next across groups. This is not super efficient. It requires correction from multiple testing when you're working with real data sets. Now, sort of just want to touch on data transformations again. This was a paper that was posted up recently on bioRxiv that aimed to assess different analytic methods that aim to deal with some of the skew and compositional bias and sparsity issues present in microbiome data sets. And I point out this one and these two because these utilize either the center log ratio CLR transformation that I mentioned or the arcsine square root transformation. And what they did in this paper was evaluate the false positive rate and the power of these methods to detect associations in microbiome data. And they found that the center log ratio method had a sort of very low power. So generally we want power to be somewhere up here around 80%. And it also had a higher false discovery rate. We generally want the false discovery rate to be below 0.05 after false discovery rate correction. Whereas the arcsine square root transformation did preserve the false discovery rate close to the 0.05 that we aim for and had a little bit better power than the center log ratio transformation, although it didn't quite, well, it certainly didn't reach the 80% power that we usually aim for, but at least it was a little improvement over the center log ratio transformation. So back to some real data here. So here's a quick example. The data set that I was working with, it had some additional variables, like the age of infants when specimens were taken, whether the infants were being breastfed, yes or no, when the specimen was taken, and also whether the infant had diarrhea in the 7 days prior to when the specimen was taken. And so these are box plots just comparing the natural log transformed Simpson diversity, Shannon diversity, and evenness measures in the breastfeeding versus non-breastfeeding infants at the time the specimen was taken. And we see that there are no differences in any of the alpha diversity measures by breastfeeding status of the infant. However, we see that there is a statistically significant reduction in the Shannon diversity in the infants who had diarrhea within the 7 days prior to specimen collection. And again, the Shannon diversity emphasizes rare species more than the Simpson diversity does. So the fact that this is different, but the Simpson diversity doesn't suggest that diarrhea may have an impact on reducing the more rare species in the microbiome. Also, I looked at age. All the diversity indices increase with age in these infants, which is expected. That's been well described. Diversity increases as during infancy as infants age. Now here's an example of ordination plot. Again, I'm using the same variables that I used to do the alpha diversity analyses. Age, breastfeeding, and diarrhea. So for this, I use a technique that is similar to principal components analyses, but allows you to use Bray-Curtis distances to perform your principal components type analysis. What it does is it creates these two axes, component 1 and component 2, that capture as much as possible the variability in the microbiome abundances compositions in each of the 448 infants. So each dot represents the microbiome of an infant, and plotting these microbiomes across these two reduced axes here gives you an idea of the differences or distribution of compositions in these microbiomes. And these two arrows are produced by the ordination analysis method that I used. The arrow going this way indicates that the breastfeeding or indicates that the composition of these microbiomes sort of tracks or separates along the y-axis component according to whether the infants were breastfeeding or not, and they sort of separate along the x-axis according to the age of the infant. So these are also color-coded here, where the lighter colors represent specimens taken at younger ages, the darker colors represent specimens taken at older ages. So you can get a sort of visual representation of how the compositions sort of spread out or distribute by age, in addition to what the arrows tell you. This is the same analysis, but now I've color-coded these by breastfeeding status. Blue being infants that were not being breastfed when specimens were taken, green being those that were. So again, we see the green specimens kind of track down towards the bottom of the y-axis here, as indicated by the arrow. This is a similar analysis, but now I've used diarrhea in this model, diarrhea and age instead of breastfeeding. And gray are specimens that were taken when in infants who did not have diarrhea in the 7 days preceding the collection date, and the black are those that did. So we see the sort of black specimens sort of tracking up towards the top of the Y component here, corresponding to the direction of the arrow, which shows that the distribution of compositions sort of varies along the y-axis in a way that reflects differences due to diarrhea. What method I was using on slide 25, what program? So this is, I'm using the package vegan in R, and specifically what I'm doing here is something called distance-based redundancy analysis. So it's sort of a principal components analysis type method, but it allows you to use like Bray-Curtis dissimilarities as the input rather than a correlation matrix, which is what you would use with the principal components analysis. So I kind of skip ahead because I have a few slides, quite a few slides left to go through, but this is just to give an idea of how you can interpret what the components mean or what they represent by plotting or looking at the correlation between the abundance of each microbe and the component scores. So component 1, for example, is strongly negatively correlated with bifidobacterium. So as component 1 increases, bifidobacterium decreases, which makes sense because the specimens separated in their compositions over the component 1 x-axis, for example. Another useful concept is the concept of community state types. So back in like 2011, this concept of enterotypes was proposed for gut microbiomes. The idea was that individuals can be grouped into subsets based on discrete configurations of their microbiome compositions, and they call these enterotypes. And these enterotypes tended to be characterized either by predominance of Bacteroides, a predominance of Prevotella, or a predominance of Ruminococcus, and were also tended to be associated with long-term dietary patterns like predominantly animal food source diets or fiber-rich plant diets. Since then, this idea that microbiomes can be separated into discrete configurations has been called into question. And it's now widely agreed that microbiome enterotypes are extremes of a continuous landscape of microbiome compositions. But the idea of using these sort of discrete groups of these discrete microbiome configurations or classifications as a way to also reduce the large amount of information in a data set to facilitate analysis is still an approach that's still used. So here is the results of a community state type analysis that I did. This was also done in R using a Dirichlet multinomial mixture model. I can provide a reference if anyone's interested, but there's a package in R to do this. And so that method identified 6 community state types in these infants. Here I'm showing a heat map of the abundance of like the top 20 taxa and their distributions across the 6 groups. There are 3 groups that are predominant, well, 2 groups that are predominantly bifidobacteria, 1 that's predominantly bifidobacteria, but also has sort of a large abundance of several other things in sort of increased diversity, and then 2 smaller groups that are either predominantly Escherichia and Shigella or predominantly Streptococcus and Lactobacillus. Yeah, so our package is called, I think it's called DMM. But I could definitely maybe share that with Laura after the talk, so that she can share it with everyone.
Yes, just email beadcore@jhmi.edu with your questions, and I'll be sure to either connect you with Ethan or follow up, however, with whatever you'd like.
So just to kind of run through, again, just an analysis of the sort of 3 variables that I was looking at in this data set: age, breastfeeding, and diarrhea. These community state types are they definitely differ in terms of, you know, the age of these infants. So the blue, green, and orange, so blue, green, orange are younger infants, or were taken at younger ages, and the red and yellow were taken at slightly older, slightly later in infancy. And the differences are statistically significant by Kruskal-Wallis test. Looking at the sort of distribution of diarrhea, yes or no, in the 7 days prior to when the specimens were taken, we see that if I sort of use the yellow group as the referent, the yellow group was sort of the most diverse group, but also the oldest group. We see that compared to the yellow group, the relative odds of a diarrhea in the blue group was significantly increased. The blue group was the group that was dominated by Escherichia and Shigella, which are 2 common pathogens that cause, or well, pathogens that cause, that are common cause of diarrhea are in those genera of bacteria. And likewise, the odds of diarrhea was increased in the green and orange and red groups, but these were not statistically significant. Only the blue group was dominated by Escherichia and Shigella. And looking at breastfeeding, using yellow as the referent, which was the most diverse and oldest group, the odds of breastfeeding was decreased in the blue group, which was the Escherichia Shigella dominant group that also had the highest odds of diarrhea. This is statistically significant, but the other groups are not significantly different in terms of the odds that these infants were breastfed. So the blue group, younger, more diarrhea, not breastfed, dominated potentially by pathogens. And so then you can also sort of take, this is the sort of taxon by taxon approach. I just decided to look at whether breastfeeding, diarrhea, and age explain the abundance of bifidobacteria using the arcsine square root transformation of the bifidobacterium relative abundance. This is a multiple linear regression model. And so the regression coefficients here say that infants who are breastfed had a larger abundance on the arcsine square root transform scale of bifidobacteria because the coefficient is positive. With each 1 month increase in age, the abundance of bifidobacteria decreased, and these were statistically significant. And then I also sort of switched this around and decided to ask whether Escherichia, Shigella relative abundance explained the odds of diarrhea. So in this model, this is a logistic regression model with diarrhea, yes or no, as the outcome. And these are the variables in that multiple logistic regression model. And so we see that the odds of diarrhea per 1 unit increase in Escherichia Shigella on the arcsine square root transform scale was 4.6 times higher per unit increase in Shigella. And this was in Escherichia Shigella, this was statistically significant. Thank you for your attention. I think there is another, okay. Any additional questions at this point? I'm pretty close to an hour, but I kind of stopped to entertain a few questions along the way.
Ethan, can you stay for a couple of more minutes in case anybody
Yeah,
has questions
certainly.
they can stay for? Okay.
Definitely.
Feel free to unmute if you have something else you'd like to share or a question you'd like to ask.
Thank you so much. That was a fantastic lecture.
Okay, thanks, Jackie. I'm glad you enjoyed it.
Excellent.
All right. And anyone who has follow-up questions, I'm happy to connect you to Ethan if you email beadcore@jhmi.edu. And thank you, Ethan. That was wonderful.
Great. You're welcome. No problem.
Bye. Thanks.
Bye.